Lecture 3 of Biology 104, where we will discuss the origin of species. We're now going to take a look at how new species evolve. There are two main types of speciation events, allopatric speciation as well as sympatric speciation. In allopatric speciation, this occurs when gene flow becomes restricted, typically due to geographic barriers. As a result, whatever population was restricted from the original population, they end up diverging genetically to eventually become their own species. Sympatric speciation occurs when gene flow becomes restricted between populations within the same area. In allopatric speciation, two populations initially become geographically isolated. Once this occurs, over time, the allele frequencies in the gene pools will change. This will cause the evolution of a new species. These allele frequencies can change commonly due to mutations, natural selection, as well as genetic drift. Eventually, this may lead to reproductive isolation as well. What is very typical is once the geographic isolation or geographic barrier is put into place, eventually, once these populations, if they do end up mixing, there's enough barriers put into place, such as reproductive isolation or behavioral isolation, that would maintain them as being separate species. A good example of this is mosquito fish that had colonized ponds that eventually became isolated. One pond had predators and the other one did not. In the pond with predators, what ended up happening is the species evolved to have a body shape that enabled rapid bursts of speed to escape predation. This was due to natural selection pressures. Without predators present, the body shape changed to favor long and steady swimming. Eventually, these two species remain separated due to behavioral selection in the females to mate with males with a similar body type. Once again, putting more measures into place to ensure that these species remain separate. Another example of allopatric speciation is looking at snapping shrimp in Panama. There are 30 different species that originally became separated by an isthmus. This isthmus restricted gene flow to eventually lead to the formation of 15 different species on the Pacific side and 15 different species on the Atlantic side. However, looking at genetic similarities between these species, it was determined that there are sister species that exist between Pacific populations and Atlantic populations, as we can see in the image to the right, thus illustrating the importance of geographic location in the development of new species. As stated previously, allopatric speciation typically results from the presence of a geographic barrier isolating a population. It is important to understand, though, that it's not the geographic barrier itself that creates the speciation event. It's the intrinsic reproductive barriers that eventually form that prevents the successful mating of the two groups after they reunite that leads to the formation of two different species. Some important trends can be noted. In general, areas that are highly diverse or have many geographic barriers present typically have higher levels of species diversity than comparable areas with less barriers. It's also important to note that as the physical distance between two groups increases, so does the level of reproductive isolation. In sympatric speciation, this is when we have a speciation event that occurs between populations. However, this occurs with populations within the same geographic area. Sympatric speciation can occur due to polyploidy, sexual selection, as well as habitat differentiation. Sometimes a new species can arise due to the incomplete separation of chromosomes during meiosis and the formation of gametes. This can result in an extra set of chromosomes. This is common in plants. In fact, over 80% of the plant species have originated from polyploidy. There are two main types of polyploidy. In autopolyploidy, this occurs when an individual arises from a single species. However, that individual will have more than two sets of chromosomes, such as 3, 4, or 5 sets of chromosomes. In allopolyploidy, this occurs when two different species produce a hybrid, and this hybrid is considered to be polyploidy. It is also considered that this hybrid can both produce viable offspring asexually. Autopolyploidy typically occurs when plants have improper cell division. This usually leads to the condition of tetraploidy, or having 4 sets of chromosomes. These tetraploid individuals can mate and also produce viable offspring. They typically do so by either self-pollinating or by mating with other tetraploids. It is important to note that these matings are reproductively isolated. This is because if these tetraploid individuals were to mate with normal plants, they would produce 3N offspring that are considered to be less fertile. In most instances, when a hybrid species is produced, most of these hybrids are considered sterile. This is because during meiosis, the chromosomes between different parent species don't pair up properly, leading to infertile or sterile offspring. However, in some plants, they can reproduce asexually. Eventually, as this continues, this may lead to the production of a fertile hybrid. This hybrid could potentially mate with each other. However, they can no longer mate with either parent species, once again leading to reproductive isolation. This concept can be illustrated in the diagram to the right. We have 2 different species here, species A and species B. Species A is considered to be diploid with 6 chromosomes, while species B is diploid with 4 chromosomes. If both of these undergo meiosis and form normal gametes, and this would then mate to produce a sterile hybrid zygote. However, if there is further mitotic or meiotic error in this hybrid plant cell, this could potentially result in the doubling of the chromosome number and the restoration of an allopolyploid state. In this instance, this new species would now be viable and would also be fertile and be able to produce offspring itself. A good example of this is exhibited in the genus Tragoponin, or goat's beard. At least 5 new plant species have originated due to polyploidy. In the early 1900s, 3 British species were brought to the Pacific Northwest. These included T. pratensis, T. dubius, and T. porifolius. In the span of only 50 years, a new species had already developed, T. misellus. It was also noted that this new species was considered to be allopolyploid. New hybrids have been formed from the mating between 2 of the parent species and also from mating those parent species with T. misellus. Recently, T. mirus has also been identified as another allopolyploid. Here we can see the mating strategy in these plants. T. dubius, and T. pratensis, and T. porifolius are all considered to be the original 3 strains that were brought to the Pacific Northwest. We can see that mating between T. dubius and T. pratensis led to the hybrid species T. misellus, noting that it is allopolyploid, having 24 chromosomes. We also can see that T. dubius mated with T. porifolius led to the production of T. mirus, also considered to be allopolyploid. Understanding how polyploidy arises in plants is very important in our current agricultural processes. Scientists are constantly producing new polyploid plant species. This is because many of these new species have higher yields, are hardier plants, or can produce larger fruits or flowers. Scientists do this by utilizing various chemicals that are known to produce errors in meiosis and mitosis. An example is colchicine, which is a chemical that is known to inhibit microtubule formation. By inhibiting the proper formation of microtubules, you are inhibiting the proper segregation of these chromosomes during these processes, thus resulting in a polyploid plant. A good example here is seen in strawberries. Wild strawberries are much smaller than our cultivated strawberries, which are considered to be polyploid. Sympatric speciation can also result due to sexual selection. In sexual selection, specific mating practices can limit gene flow between species that occur in the same geographic region. An example of this would be cichlids in Lake Victoria. Lake Victoria has an extremely high diversity of species. There are over 600 species that have evolved recently, just over the last 100,000 years. And this resulted from the radiation event of a few ancestral populations. There are two main theories that have been proposed to explain the species diversity in Lake Victoria. One is that perhaps these species are adapted to different resources, much like we saw in Darwin's Galapagos finches. Another theory is suggesting that the speciation and diversity is due to female mate selection. The following example demonstrates the importance of sexual selection in cichlids. Research has examined the mating strategies between two similar species, Pandemilia pandemilia and Pandemilia nereri. Two light conditions were studied, normal light conditions as well as orange light conditions. Under normal light, the species appeared distinct from each other. In this type of environment, females were able to discriminate and showed a preference for mating with their own species. However, under orange light conditions, the species did no longer appear distinct from each other. In this type of environment, females were not able to discriminate between males of their own species and did not show a preference. As a result, hybrids were produced, and these hybrids were considered viable as well as fertile, thus suggesting that these two species are not genetically diverged enough from each other yet. However, this provides a unique insight into how the speciation process occurs. Habitat differences can play an important role in the formation of a new species. New species can evolve when subpopulations utilize different resources or habitats that are not used by the parent population. This can be seen in the evolution of the apple maggot fly, Rhagoletes palminella. The original host plant in the ancestral population was hawthorn trees. However, approximately 200 years ago, European settlers brought apple trees and planted them across the landscape. Because these flies in the Rhagoletes genus have a tendency to mate on or very near their host plant, this innately created a prezygotic barrier of habitat isolation between populations. Thus, the populations that decided to feed on apples were isolated geographically from those that fed on hawthorn fruit. It is also important to consider that apples develop relatively quickly. This means that natural selection favored apple breeding flies for faster development, thus creating temporal isolation as well, which led to the rapid evolution of this fly.